Okay, we might make a start. I'd like to begin by acknowledging the traditional owners and custodians of the lands from which we meet today, the Wurundjeri people of the Kulin Nation. I'd also like to pay my respects to their elders past and present, and elders from many other communities who may be present today. It's my enormous pleasure to introduce today's speaker, Stefano Mangiola, who's a senior research officer in my lab. Stefano did his undergraduate degree and Masters of Science in Milan, and followed that by coming out to Melbourne and doing a MPhil on parasitic helminths in Robin Gass's lab at the University of Melbourne. Then he began his PhD on the investigation of prostate cancer microenvironments with Chris Hovens and Jeff McIntyre, and was based at NICTA right before it disbanded. So I first learned about Stefano when he and another PhD student were left isolated and abandoned in the Department of Neurosurgery at the university, and he came over here and sat in my lab. He was awarded his PhD in 2019 and then joined my lab formally as a postdoc. His research interests lie in the role of the immune system in cancer control and as a tool for prognosis. And his general approach is really interesting. It combines computational models, working on single cell data, spatial omics and other big data sets. And unusually for someone from the Bioinformatics division, he has been known to generate his own data. He's been hugely productive, both in terms of methods development and publications. And Stefano's really challenged me with his ability to introduce archaic English words that I have never heard of into his manuscripts. I hope that today he will avoid archaic English words, but there may be one or two. Today he's going to be talking about recent work developing a whole body map of immune cells. Stefano.
All right, thanks, Tony. Definitely the best introduction I had so far. So today I will present a recent study that is the whole body map of immune cell composition and communication. So just because I haven't never spoken to this audience, I introduce where this study sits on what I generally do under the umbrella of computational cancer immunology. Part of my research is driven by biology, and I studied prostate cancer and more recently breast cancer. These half dots are the studies that are ongoing, and some studies about the immune system, so focused on the immune system, but targeted on cancer. And also I do some technology development among statistical methods for mostly the convolution and composition analysis, and some more basic models for RNA sequencing, and also some analysis infrastructure and visualization. And the study today, only for that, will involve this immune category as also the use of a model we did on for compositional analysis. So also with the help of modern technology, we are going more and more to gain a holistic perspective of biological system, for example, the human body. And this is a simplification, but the paradigm that we use is often to study a biological system or subsystem, for example, liver, use some technology such as a mouse model for a goal, let's say standard cancer evolution. And you can imagine this can be applied to many tissues of the human bodies across different technologies with different focus. And then we publish studies and we build up a literature body. And as a researcher, we build a mental map of all this literature. This map has spots, of course, many studies as a specific focus, but nonetheless, we are able to make sense of this, understand the underlying system, and go on with further studies, either for filling these gaps or translating some of the discoveries in useful tools. So more recently, another paradigm is emerging, where we can take all the subsystem. Let's talk about the human body, probe them with a scalable and consistent technology. And one of the most scalable technology that we use now is single-cell RNA sequencing. And we are able to collect thousands of samples across hundreds of studies. And at this stage, that we can build a quantitative map of the whole system and use this map to then study specific phenomena such as cancer, basic immunology and biology, development, aging, infection, and so on. And then from these findings, we can follow up with validation or further experiments. So building the map as, in a way, shifting the position in the pipeline that we can use. And this is quite obvious when you think about really recent developments in artificial intelligence, when especially ChatGPT, that we all know, was able to integrate books, web, and internal sources to build a map of the human knowledge. And so rather than having an expert in laws, expert in communication, and so on, intelligent systems are able to process this quantitative map, and then we can extract domain-specific knowledge from this. And we can imagine we could do the same, collecting hundreds of millions of cells across sources, creating a human map that is compatible with intelligent systems, so machine learning can learn from it, and we can go toward the next generation personalized medicine, where we can imagine we could profile individuals even before they come to the clinic, as we will acquire quite robust knowledge about what the immune system of a female of 42 years old of a certain ancestry could look like. And so this is quite fundamental and important, and there are a lot of potential fields that can be impacted by this, but just a couple of examples. For example, precision medicine uses the patient characteristic to provide better treatment and diagnosis. And you can imagine the power of understanding the heterogeneity, for example, of the immune system or at the tissue level to guide precision medicine research, and even have a quite good understanding of the characteristic at the cellular and molecular level of an individual even before it comes to the clinic. Also, immune therapy has obviously been extremely successful, for example, in cancer. And each of us has a different immune system and immune history. So it's quite important to understand what's the heterogeneity and what are the trends that exist within the population. Even before this, again, to guide research, but to have a clear understanding of all the boundaries that the immune system, for example, can take even before seeing a patient. So the aims of this study was to use large data to model a map of the healthy immune system across tissues, quantify the heterogeneity of the immune system across demographics, and share this map and data with the community. So before to do all this, an important step is to take several sources of data and harmonize them and curate them in a way we can analyze them all together with the same statistical model. So what we have done is to take, there is a big repository called Cell by Gene of single cell data. And we took 30 million cells from this repository. At the time, it was January 2023. And these data exists across more than 45 tissues. So we have harmonized it, curated in a way I will describe, and created an interface that allow even non-specialists to explore this data, filter the cells of interest, for example, CD4 T cells across tissues for young individuals, let's say, and then collect these data for analysis. And also very recently, Cell by Gene infrastructure also published his exploratory tools that is somehow similar to this. And is now fairly straightforward with few lines of commands, in this case, to take your favorite genes, your favorite cell types, a plot for every cell, the gene expression here on the y-axis across disease, let's say. and this is on the x-axis. And here is color by data set. We can start to understand the heterogeneity that is present. Or we can do it across tissue, again, appreciating the heterogeneity that exists. Or we can quite easily quantify the immune infiltration at the tissue level here on the x-axis, and understand what are, for example, the organs that tend to be hotter or colder, and appreciate the heterogeneity we have within a tissue. So I will spend a couple of slides to show you what's the content of this big database. It includes several, more than 10,000 samples. More than 700 are from prenatal or toddler, 1,000 roughly are from pre-adulthood, and more than 10,000 are from adulthood. Again, more than 45 tissues represented here, and it's worth mentioning that this huge atlas, multi-atlas level data set is quite complex in structure, as you can imagine. So we have tissues, and each tissue is generally contributed by more than one data set. Let's say 5 laboratories did a study on heart and contributed to this database. And each data set is contributed by multiple samples. So first of all, we have a hierarchical organization here, and then each sample has a more simpler, if you will, data structure where we have each sample as a property such as ethnicity, age, sex, and technology. So this is quite important when we want to model this data to be able to fit these complex structures. We did an analysis of the metadata and the content of this Atlas level database. And we observed, of course, this resource has not been done with a specific design. There are some biases and some gaps that is important to acknowledge. For example, if you plot each dot here is a sample, and the color yellow represent many data sets contributed. We can see that the first on the x-axis we have disease, and the first column is healthy tissues. We can say already for healthy that this data set is skewed, as we might have imagined, toward European ancestry. But this bias becomes even stronger when we look across diseases. So there is this huge gap. Where for some ethnicity, we have no samples relative to a certain disease. Also, age is a very powerful, powerful data source in this data set, as we have.
He might have a YouTube
is it good? Yes.
Something that needs to be turned off, like something in the background. So, and people at home can hear it as well.
Is something more on your computer on the computer?
Anyway, so we have a broad age representation from 0 years to 84 and more years old. However, the age representation is not the same across dataset groups, such as ethnicities. We can see here that some ethnicity has a very age coverage, some others not so much. Also tissues, this is true. We can imagine prostate sample might be sampled later in life rather than blood. And also this is important for the statistical model, as this grouping, they have data coverage that do not overlap. Okay, so the last part about this introductory section is about data curation, as these studies focus on the immune system, and you can imagine every study has a different standard for cell annotation, also different nomenclature. So we did a quite big effort to harmonize and curate cell types along a common differentiation hierarchy. So what we did is to take the original annotation, give a common ontology, and then took these as a source of evidence, and also we annotated each cell with other 3 methods, including single R with the Monaco reference, for who know what it is, blueprint reference, and Surratt-Azimuth reference. And so we took these 4 sources of information and we identified consensus. So we identify consensus tiers. For example, tier 1 is where all sources of evidence agree, and this is likely the majority of cells. and so on for when some of the more than one of evidence do not agree. And for the study from here on, we have eliminated confidence tier number 4 to try to keep the reliability of our study high. It's also worth to observe that the distribution of confidence across data groups is not constant. So yellow represents the first tier. And so we can see that across technology, there are some technology for which the reference-based annotation do not often agree. This is important to note, and also, for example, across ethnicities. Then we are not sure if this is the reason, but we can imagine that these references have been built with specific technologies and human samples coming from maybe more some ethnicity than others. So this is also an important point to note. And lastly, we ask ourselves, which are the cell types that are harder to distinguish? So when all methods do not reach consensus, which cell type that co-occur normally? And so on the y-axis here is the counts of cells, and the cell type combination on the x-axis. Of course, you might not be able to read, but I will quickly point few things. So we can see that the blue colored represent cells that have common lineage. And of course, these are generally harder to distinguish. We can see CD4 central memory from CD4 naive, CD8 effector and central memory, and so on. But some of the cell pairs that do not have a very common origin, such as NK cells and CD8 effector memory, as they have similar function, the transcriptome tend to converge. They are also very hard to distinguish, and so we show these rank as help to guide possibly further efforts with the reference methods to where we should focus on to improve this reference-based annotation. So I will spend a couple of slides now that we have our data set. How did we extract information and knowledge out of it? So big part of this study is to model the immune composition throughout the body. And so we developed this method that is called SCComp and is a compositional statistical model for single cell data. And I presented this before, but for this study, we have extended this method to atlas level analysis. So we extended to multi-level modeling, able to analyze complex and hierarchically organized data set. So what is compositionality, for example, for the immune system? It's quite straightforward. Sure you have seen a UMAP before. Each dot represents a cell, and cells are closer to each other when they are similar transcriptionally. We can cluster similar cells, and then we can count for each cluster how many cells do we have. And for example, here we have 10 samples on the x-axis, and on the y-axis the counts. As you can see, some sample might have a lot of information, a lot of cells. Some other samples not so much. So the first thing that our method does is to model the data with the noise that is explainable by counts. So this is quite specific. But then we cannot model counts directly. The meaningful information we can model from those are proportionalities. So you can imagine that these counts underlie proportions, and when we plot them, we can start to understand, for example, between two conditions, stable and progressive disease, whether there are some increase or decrease of, for example, CD4 T cells across samples. Another thing that we did, and we described with others, Belinda is here as well, that the relative abundance of a cell type, let's say 40% of T cells, and the spread around this 40% is not independent, meaning that more abundant is a cell type and more variable it is. And so when if we plot this, of course, is a cartoon. If we imagine that the red square will be the reality, so we have a consistent spread or variability across cell types based on their abundance, we found that the reality is not this. So we have rare cells that tend to be, relatively speaking, less variable than abundant cells. And in our model, we model this in a logit-log space, which is anyway an unconstrained space where the statistical model often work in. And we can identify very often a very straight linear relationship between these two quantities. And so because our model is a Bayesian model, so model all this variability in a joint way, we are able to very effectively transfer information across cells types. So each dot here is a cell type. And lastly, we observed that this type of data set often includes outlier observation, which are observations that are not explainable by the model we use. So sometimes it's parsimonious to identify them and exclude them from the modeling. And so our statistical model is able to do that as well. So this has been published in that preprint, but what we did now is to extend this model to be able to analyze more complex data sets. So we are able to do multi-level analysis, and what this means is that instead of imagining that all data points, let's say samples here, belong to one group, like in the top, we can model them belonging to different groups, so we can do a hierarchical inference where we infer the association for each group, and we can ask also more complex questions overall. So we can ask overall, do this group tell the same story? In a way, they are all decreasing. And so this model become more powerful and can convey more information. Not only are more powerful, but are also needed in some case. For example, if we have two groups and they don't overlap in the x-axis, we saw the case of prostate tissue being, mostly old individuals, while other not so much. We could have this Simpson paradox where if we don't consider groups, we have one association, let's say negative, but this becomes positive when we consider data belonging to multiple groups. And so I will show you analysis afterwards, but just to give you a picture, what does this mean in our data? Let's say we want to test how B memory cells increase or decrease in relative abundance as we age. So from 0 to, well, not 0 here, but 1 here to 84. And we group this data based on tissue. As we see, first of all, we can see quite a lot of heterogeneity. And we can then ask per tissue what's happening, but also if there is a body global effect. So again, we can ask questions hierarchically here. And one thing that is not often talked about is that when some groups, let's say blood, contributes with much more data than others, let's say here, I'm visualizing where blood is the first rank on the top plot, contributes almost 10 times to the second rank, which is kidney here in terms of samples. The story we would extract will be mostly about blood, while other tissue will be completely covered by this information. And this is not so much the case if we consider groups. Anyway, so I've described you what's the data set and what's the model we use. And now I will describe how we use these two to build the whole body immune map. The pipeline is is pretty simple in a way. We take our data set and we model two quantities. One is immune cellularity. So we have 40% of immune cells in a sample, and the other one immune composition. So forgetting non-immune cells, what's the composition of immune cells across cell types? We model, and we model effects such as age, sex, ethnicity, and technology, and we group the data with the factor I just mentioned. And we do hypothesis testing, let's say, is cellularity increasing or decreasing? And we are able to remove unwanted variation using this model to be able to visualize the data and extract the information out of that. For one thing we can do, for example, is to start to estimate profiles of the demographics. Let's say we want to estimate what's the mean immune composition across the body for an individual's, here we have average male and female effects, of 42 years old of European ancestry. And so we can get these reference maps. In this case, these are healthy tissues. And so how this type of demographics would look like across cell types on the x-axis and tissues on the y-axis, and the cells represent proportionality. I don't have time to go too much deep into this, but you can imagine the application of having a reference baseline whenever you try to understand a case where the homeostasis is lost. It's very important to have a very robust baseline derived from a large amount of data. And we could cluster tissues based on their immune profiles in terms of composition, identifying lymphoid tissue signature, lymphoid-like B-cell enriched clusters, Treg and T helper enriched cluster, and so on. So that's one of the outcomes we show in the paper as soon as preprint. One other thing we have focused on is aging. We are able to do hypothesis testing and ask questions to the model. For example, does immune cellularity increase or decrease as we age? So age-related inflammation is a well-studied phenomenon, and the concept is that as we age, we acquire inflammation on low level, but a chronic inflammation, and this is important for autoimmune disease, cancer, and so on. So it's very clear that the inflammation is, as we age, involves gene expression, so the phenotypes of immune cells. But it's not so clear what role it plays, the fact that the mere amount of immune cell in our body might increase in concentration compared to the non-immune cells. And so we could quantify a 1.2% increase of immune cells as a global effect in our body every 10 years, which is quite substantial. We went beyond that, and as I mentioned before, now we can ask questions about the tissues. So is there a consistency across tissues for this immune accumulation or not? And we found that although most tissues, they have this immune accumulation, some, such as skin, intestine, and heart, do not. So actually, there is a depletion as we age. And this is also quite important to understand for, have all sorts of application, for example, cancer and immune therapies. We went beyond immune cellularity and tried to understand the immune composition. So does the immune composition changes as we age? And the first plot represents effect at the body level, so above, what story all tissues together tell. And we identify quite common pattern, well-known patterns, such as one of the biggest depletion of immune cells, relative depletion, is about naive lymphocytes, and this is well known as we grow with the repertoire of naive lymphocytes that then get depleted as we grow and we deal with infections. But interestingly, we also identify mast cell with patrol our local tissues as decrease as we age. On the other hand, we have the most contributors to this immune accumulation are monocytes. And also we identify T helper cells, although not all significance. You can see the threshold on the horizontal line, but definitely almost all of them, they follow the same pattern. So altogether, the pattern is quite striking. And you can see here the same representation, but where we remove the unwanted variation, we just visualize the effects on the data, and each dot is a sample here. Again, we can increase our resolution and build a map of immune changes across the tissues as we age. And so we can start to understand, do all tissues age in the same way in an immune context? Again, here there is quite a lot of information, but I will just point to a couple of aspects. Again, the cell types are on the x-axis and tissues are on the y. The black dot represent significance, and we have red for increase and blue for decrease. One thing we observe is that Again, it's known that thymus is very important lymphoid organ, and the thymus ages with us. And so there are important changes. But we definitely identify thymus as the first, the most altered tissue as we age, above all other lymphoid organs and also non-lymphoid organs. And one quite interesting finding is that the greatest depletion of relative depletion of immune cells is about T regulatory cells, above all others. And this tells the same story. So on one hand, we have immune accumulation, mostly monocytic, but also about antigen-experienced T cells. And on the other hand, we have a depletion in numbers of T regulatory cells which regulate our immune system in the thymus. And other important tissues in this context are liver, uterus, intestine, and so on. And quantifying the cell types that have more often significant alteration, we identify macrophages, again, monocyte, B memory cells, and other lymphocytes, and so on. So you can imagine now we start to gain a more holistic view of the immune system through our body, thanks to these new data sets and data modeling techniques. So in this study, we also try to ask, okay, beyond this merely compositional alteration, what are the qualitative, if you will, changes, for example, in terms of cell-to-cell communication? And so for some of the tissue that had enough information, as you can imagine, communication is a harder effect to probe from the data. So we were strict on the information amount we were modeling. So for example, across these 4 tissue, following again T regulatory cells in blood and lymph node, we could see that there is an alteration in communication. This is again a volcano plot. And there are some axes of communication. So each dot is an axis of communication, which include few receptor and ligands that are decreasing in strength and increasing in strength. So there is not a global depletion effect in this case, but definitely an alteration. Many of the depleted communication axes involve the basic function of regulatory cells, but also some of the enriched axes of communication, they have interesting pattern. These are 2 of them. For example, although they generally increase in strength, they mostly increase in strength between T regulatory cells and naive T cells, while they decrease between T regulatory cells and antigen experience, which obviously is a very important interaction to regulate them. So again, this tells us that not only we have a compositional effect, but also effect at another level. Going on with communication, this is a more landscape analysis, and we have analyzed, here we're plotting in the first plot, each dot is a communication axis that was significantly altered. And on the y-axis, we have cell types, and on the x-axis, effect. So one overall effect at the body level, we can see that we have a list on top of cell types for which the communication, the significant alteration are mostly decreasing. So there is depletion of communication, while it's not true the other way around. So we can see that overall we have more an effect of communication depletion as we age. And the top 2 cell types are macrophages and dendritic cells. So 2 important professional antigen presenting cells. And here are the volcanoplot that represent those 2 cell types. And in this case, yes, we see a quite dramatic bias toward communication depletion across many of communication axes that are important for migration and antigen presentation and clearing. Again, going across some of those communication axes, here we can see between dendritic and macrophages and which cell type this communication is mostly disrupted. Should I mention that blue represent depletion and red increase. And so we can see that, for example, dendritic cells in blood for some key communication axes are depleted toward a lot of other cell types, including lymphocytes and myeloid cells. For some, communication axis is mostly depleted among myeloid cells, but you can see here the picture. So the communication depletion is very spread across cell types. And this analysis was done with cell chart. So broadly, the picture we could extract, the landscape we could extract from this analysis about the inflammation related to age was that few different mechanisms play together. For example, we have, as I said, an increase in the relative amount of monocyte cell through our body. At the same time, we have a depletion of T regular thymus, so we have a lower regulation. And also, antigen-presenting cells could have disrupted antigen clearers, as far as we can see with the communication depletion. So these go toward inflammation. Now, antigen clearance can contribute to inflammation, and as antigens sit in the tissue can stimulate other parts of the immune system, also might affect the immune plasticity. So, and also some pathways that were linked to migration to lymph nodes. So in a way, these two mechanisms playing together as we see the immune accumulation in our body, but our immune system becomes less targeted and less adaptable to new stimuli. So this could explain a background level of inflammation that is increasing that is not specific. The last couple of slides, I will show you other aspects we describe of this immune map, which is the heterogeneity across demographic groups. For example, is the immune system in terms of composition different between sexes? So we could not identify a significant difference in terms of abundance of immune cells and the variability. So the two cohorts, male and female, seem to be consistently variable. When we go to immune composition, we could find some significance, although not huge effects, about the depletion in the male cohort compared to the female of cells such as megakaryocytes and immune progenitor, an increase in lymphocytes such as CD4 helper T cells, but not much more than this. If we compare ethnic groups, here we have compared each ethnic group with the average or mean of the rest. And here we see a richer picture. For example, for relative immune abundance overall in our body, we see that, for example, African and European differ as Africans show a less abundance of immune cells overall compared to European, and this is the data showed that of which infer this effect. As you can see, European in the middle there. Each dot here is a sample, and we are plotting the immune proportion. If we go to tissue-specific effects, again, we can identify some of them. For example, African have a depletion mostly in heart tissue and kidney, while European take into account all other biological and technical effects, of course, have an increase in breast and heart. And also we identify, I won't go too much deep into it, but at the immune composition, again, we can identify difference between, for example, the Asian cohort especially and all the rest. So while I won't focus on specific finding here, because of course these results about ethnicity should be taken with care, as I just mentioned, that some ethnic group, they have not a huge data coverage. Although the broad picture is that we provide evidence for heterogeneity across ethnicity, and together with the observation that we have a strong bias of data production for some ethnic group, is very important if we want to develop a ethnically balanced precision medicine research that will be applicable to everybody in the world, to really have a thought about where do we get our data from, and maybe prioritize some part of the demographics. So to conclude, we have harmonized and curated 30 million cells shared through our interface curated Atlas query. We have, in 45 days, the community has been using this resource quite a lot. We have shared more than 3,000 gigabytes so far, and this is going up. We have developed a statistical model for atlas-level compositional analysis of single-cell data. We built an immune compositional map of the human body and used this map to do two things mainly: resolve age-related immune changes at the body and tissue level, and resolve heterogeneity across the demographics. Thanks for the attention, and let me thank some of the institutions that supported this project. A lot of people from Tony Pfaffenfuss group, Terry Speed, and also from other university overseas, and the Bioconductor core team also contributed quite substantially. Okay, thanks a lot.
So Stefano has left us plenty of time for questions.
Thank you, Stefano. Just an incredible amount of work. Congratulations. The question I have is about whether you have taken into account the method of dissociation for the different tissues, because this is really key in terms of the immune populations you collect, the efficiency of which varies dramatically in terms of representation of cells that are there or not. And probably has also changed a lot through the history of single-cell genomics. And one sort of detail on that stood out to me is your observation for mast cells being captured in younger populations. Mast cells are famously hard to capture by a single cell, and I wonder, after the initial years of the Human Cell Atlas focusing on adults, now looking at children with better technologies for isolation, whether that could explain. Could you just elaborate on that? There's a lot of things I've mentioned, I'm sorry.
Yeah, no, it's pretty clear. Yes, so that's a very fair point. So we did, for example, we took care, first of all, for when we take data set for immune cellularity, so how many immune cells are in a tissue. We curated, William is here, a lot of samples and data set to make sure that the protocol was not enriching for immune cell or depleting by design. That's something we could control. Now, the point you make is good. There is a limitation in a way. This is very huge data set. It's hard to check. I mean, there are more than 500 studies, and to model these aspects, although a discussion is helpful. I mean, I would say some effect, if there are very huge effect that cannot be noise, but there are huge trends also in terms of time, this could be important. But the hope is that on these very big meta-analysis studies, many of the sporadic effects, depending on one laboratory or another, they get pooled, and still we are able to extract signals out of the noise. But it's very important, of course. Yeah.
I'm going to go to Teams for the next question. This is a question from Andrew Roberts. Thank you for a stimulating seminar, Stefano. How can you test the hypothesis arriving from your analyses regarding specific inflammaging effects and communication gaps?
How? Well, as I said, we use CellChat, and what this algorithm does is to, for each sample, separate the cell types and understand how co-expressed ligand and receptors are between all cell type pairs, and this is the inference. Then we use BRMS to model the data. Again, we use multi-level analysis. And so on one hand, the CellChat tells us, this communication axis has this strength. And so we have thousands of samples, and we, again, try to understand the effect, whether the overall communication axis strength was increased or decreased based on age, in this case, age. And we did it, again, at different levels. overall effect at the body level and the tissue level as well. I hope that answered the question.
And in case he meant validate, could you comment on how these things that you're identifying might be validated experimentally?
Yeah, for sure. Models can be used. Model of aging could be used to validate this mechanism at the expression level, but also with other experiments that goes beyond gene expression. Probably this. I mean, yes, this map offers streams of research and validation. So probably I can imagine, choosing communication axes of interest relevant to a disease, for example, and validate those,
Yeah, I have similar sentiments, and it's a huge amount of work and really valuable. But it also in line, I guess, with Hamish's question. I couldn't see neutrophils anywhere. Can you comment on how you think the technologies which are missing them are impacting our interpretation and all of your networks of communication? Yeah.
Neutrophils are another cell type that is somehow challenging to probe and is not always probed. However, neutrophils are in the study. For example, they appear to be to change in relative proportion. Let me see where they are. Are they here? No, actually, probably not. I thought they were included in some analysis, but I mixed them with some other cell type. Yes, I mean, neutrophils are challenging to probe, and we might not be able to tell a complete story for every cell type. I mean, I'm not sure what we can do about that. As we increase the number of data set and integration, we increase the power of knowledge generation, but also there are some limitation on how much we can control. So I don't have any specific comments, not to say that, it's, again, I'm aware of this, and it is worth.
I'm going to go back to Teams for the next question. Joe Groom asks, following from Hamish's questions, do you consider the timing that samples are taken? Circadian rhythms play a large role in cell migration, cell interaction, and the makeup of lymphoid organs. So what's going on with that?
Sorry, with the timing at the samples were taken, so the years, for example?
The role of circadian rhythms.
Circadian rhythms, oh my God. Well, I mean, the key that a piece of information should be available across 10,000 samples to be, or the majority of samples to be able to model this. I could model these broad effects because age is ubiquitous and sex and so on. I'm not sure how many, again, not many studies would have recorded information about the circadian rhythms. I mean, that would be amazing. Maybe we want to collect that information across thousands of papers, but we cannot. It's dependent on the information we can harmonize at the large scale. So that is missing, for example.
Hi, Stefano, great talk. I was interested in the way that you analyzed your placental tissue, for instance, from the cell by gene database. Placental tissue?
Placenta, yeah.
Because in the cell by gene, obviously, placental tissue is a mix of mom and baby. And majority of the immune cells that you'd get would be, maternal and fetal, depending on where. Did you make that distinction? Because they would have very different signatures.
Yes. Well, we also often grouped tissues together because there are hundreds of tissues, actually. So to be able to understand interpretable patterns with the confidence, sometimes is good to group. Of course, the level to which we group must be critical in some specific fields. The answer is that if these two samples, I will check, but if these two samples are both present, we group them under the placental name. I mean, I will have a look. This is
So
also
one of the things I noticed is that you had your placenta as 0 years old. Right, one of the first slides, which means that any maternal immune cells there, you wouldn't be counting those as.
Yeah. I mean, it's worth to take those things into account. For example, these will be taken into account. It's a fine balance between separating too much and too little, but there are major effects of which I'm not aware of, because we have basically the whole body map that will be worth taking into account. So maybe we welcome if you're field specific and organ specific, email us, and thanks for the heads up. Yeah.
I have a question. That was a really impressive talk. Thanks, Stefano. About how many cell types do you find by integrating all these data? Can you answer the question of how many cell types are in a healthy person?
So we did not integrated the data per se. If by integration, we mean project them in the same space. As you can imagine, tens of millions of cells, it is very challenging. We use the strategy of analyzing the information related to each data set independently, and then build a model of that. So that's as a premise. The cell, and also we focus on immune cell types because they have a quite standardized ontology. There are many immune, there are close to hundreds cell types that are being classified. But if these are not consistently classified across samples, they have no utility. So that's why we have summarized these, I think must be close to 50 immune cell types across, between 20 and 30 immune cell types. So we took decisions, again, on the resolution. So yeah, there are a lot of cell type in the data set, but have to be harmonized for them to be helpful.
Yeah, great talk. Lots and lots of work. I'm going to ask a bioinformatics question. I noticed, obviously, there's so much detail that you have to skip over in these types of talks, but you talked a little bit about batch correction of the compositional data. Mm-hmm. And I was just wondering if you could give me a flavor of, just an idea of how you did that, because I can imagine it be really challenging because you have limited number of cell types. So usually when we do batch correction on RNA-seq and we do RUV, we've got 10,000 plus genes. If you used RUV, I think you did say you used RUV. So I was just wondering how you go about doing that on that kind of data. Yeah. And with the batch effects, are you able to attribute what is driving the batch effects? Like what is the main driver of the batch effects? Because there's so many technical factors here.
Yeah. So we did not use RUV. We use our own model to remove the unwanted variation. When I say unwanted variation, I mean known unwanted variation. It's not like RUV where you remove possibly unknown unwanted variation. So in this case, we use residual methods where we decide, basically, you feed the data with your covariates. You decide for which covariate you want to preserve. We calculate residual, and one of the beautiful things about this Bayesian infrastructure is that then you can generate the data. You can ask the model on the joint probability distribution with the effect you preserve to generate the data according with the residual you have. We use the same model to remove the unwanted variation, but is known, is just what you decide you include in the model. Yeah.
You showed us cells and cell number, but what about function? Do you think you could ever extrapolate to function? Because unlike what you showed with sex, where there was apparently not very many differences, which I found interesting, but with function, there is a difference in bisexual dimorphism.
Yeah.
There's lots of evidence for that.
Yeah.
But how would that come about then?
Yeah. So of course, sexual dimorphism is a very known phenomenon. I have to specify these are, just to put it there, are healthy, just healthy tissues, but of course, difference exists. Yes, I mean, about the function, it's for sure is an extension, can be, but is a natural extension of these studies where we go more into the gene expression and pathways that are expressed. So of course, tackling everything in once is, it doesn't make sense, it's too huge. So this was the first step. So then going beyond composition and toward more qualitative or functional aspects is important, and yeah, it will be done for sure, yeah.
It's 1 minute to 2. Hamish has another question, but maybe he can ask after we've finished. It just remains to thank Stefano again for a very good talk. Thank you.